In this video, we're going to talk about what a cell is. So a cell is the lowest possible indivisible building block of life. So you can have single-celled life forms, and you can have multi-celled life forms like humans, which contain trillions of cells, but you can never have a life form made out of anything less than a cell. The individual components of a cell can never in themselves make a life form. So what is a cell? At its most basic, a cell is a sphere. It's like a water balloon. It has a membrane that prevents water from passing. So you have water on the inside of the cell, and you have water on the outside of the cell, but water is not able to pass through. And this is because of the way the membrane is structured. So if we zoom in on a little segment of the cell membrane, we'll see that it's organized like this. It's made up of a type of molecule called a phospholipid, which stylistically tends to be drawn something like this. And the phospholipids connect together and form a structure called a phospholipid bilayer. So each one of these little round balls with two tails is a phospholipid. And this ball is the head. And the head of the phospholipid is polar, which means that it has electrical charge, and it will interact with other objects like water molecules that are also polar. So water will tend to be drawn to the head, and the head will be drawn to water because both have electrical charge. Then we have the tails, and the phospholipids in cell membranes generally have two tails, and the tails are made up of fatty acids. And fatty acids are non-polar, which is to say they're hydrophobic. They don't interact with water, and water avoids and actually repels fatty acids. And this, since you have water on the inside and on the outside of the cell membrane, will cause the heads to push outwards in each direction and the tails to push inwards. And this tail layer, since it's unpolar and hydrophobic, will repel water in both directions, so water can't move through. Okay, next we're going to talk about what we find inside the cell. And the cell is primarily water. About 50 to 70% of the cell's content is water molecules. Then the second biggest component of the cell is protein. And proteins are little machines. They do all kinds of things inside the cell. All the cell's different functions are performed by proteins. So if we zoom in on the inside of the cell, we'll find lots of little proteins. And one of the most common proteins is enzymes. And enzymes are usually stylistically drawn like this, like little Pac-Man. And the purpose of enzymes is to increase the probability of a certain chemical reaction happening. So they decrease the threshold at which the reaction happens, which makes it more likely to happen. And in this way, the cell is able to drive chemical reactions in the direction towards the equilibrium that the cell wants. And they do this by getting objects stuck inside their active center, which is like the Pac-Man's mouth, and then somehow changing the object that gets stuck in there. So that's an important type of protein, but proteins fill lots of different functions inside the cell. Another type of protein is the structural protein, which makes up the cell skeleton. And if you look inside the cell, you will see long strings like this. And they're made up of proteins that self-form into these long strings, and they help the cell to maintain its structure. For example, underneath the cell membrane, you will find long strings of proteins called actin that help the cell to maintain a correct shape. That's the second type of protein. A third type of protein you will find is the membrane transporter, which is a protein that sits in the cell membrane and that transports things into or out of the cell. Because obviously, if if you just had this phospholipid layer and nothing else, nothing would ever be able to pass through, and the cell would not be able to do anything. For the cell to be able to fill any kind of function, things like ions or molecules need to be able to pass through the cell membrane. So you find structures like this, that are proteins that will, for example, allow ions to pass into the cell. So that's the third type of protein. A fourth type of protein is the gene regulator. And to talk about the gene regulator, we'll first have to talk about DNA. So after protein, the next most common component of the cell membrane is nucleotides. And nucleotides are molecules that, among other things, form DNA. And DNA is information. It's used to store information about how the cell will be structured, how it will behave, and DNA is found inside a structure called the cell nucleus. So the cell nucleus is like a round ball. You can see it even when you look at the cell in a regular light microscope very easily. And if you zoom in on the nucleus and zoom in on the DNA, you will see that the DNA is a long string. And on this string, attached to it, you will find all kinds of different proteins with all kinds of different shapes. And these are the gene regulatory proteins. They decide when a certain stretch of DNA is going to be copied for the purpose of creating new proteins. So everything in the cell happens as a result of proteins doing it. And the way in which the cell decides what type of cell it's going to be and how it's going to behave is by creating specific types of proteins on command at specific times. And this in itself, which can be kind of confusing, is determined by proteins that are attached to the DNA. So we have lots of different types of proteins that fulfill lots of different functions. We have enzymes, we have structural components, we have membrane transporters, and we have gene regulatory proteins. And if you zoom in on the proteins, look more closely at them, take this one for example, we'll see that the protein is actually a long string. And this string gets a certain type of three-dimensional configuration that allows it to fulfill its function. And the string is made out of molecules called amino acids. So a protein is a molecule made up out of a string of amino acids. And if you zoom in on the string, you will see that there are different types of amino acids. Actually, there are 20. And each one has a side chain, which is a little part that sticks out of the side. And each side chain has its own specific behavior. It has a shape which causes it to have a specific polarity. Some are non-polar, some are charged, some are acids, some are bases. And this causes the amino acid to interact with its environment in different ways, depending on which amino acid it is and which side chain it therefore has. So the specific sequence of amino acids causes the protein to gain a specific three-dimensional conformation. And that three-dimensional conformation in turn determines how the protein functions, what function it fulfills in the cell. So anyway, getting back to DNA. So DNA is a long chain that contains information about proteins. And if we zoom in on this chain of DNA, we will see that it's actually two strings that are parallel with each other. And on these strings, and these strings are made up of molecules called nucleotides. And when it comes to DNA, there are 4 different types of nucleotides. We have thymine, we have adenine, we have guanine, and we have cytosine. And so these are the 4 different nucleotides you can find. And DNA can have any sequence. And in general, since DNA codes for amino acids, what you will find is sequences made up of triplets of DNA. So we'll add another nucleotide here to make a complete triplet. So this is a triplet, and a triplet is known as a codon. This is an important term. And one codon in DNA codes for one amino acid in a protein. Okay? So, and since there are 4 different nucleotides, that means that one triplet, or rather, there can be 4 times 4 times 4, which equals 64 different possible triplets. So there are 64 different possible triplets. Here we have TGA, for example, which will code for one specific amino acid. And since I said before that there are only 20 different amino acids, that means there are multiple different triplets or codons that might code for the same amino acid. But that's not a problem. The only thing that's really required is that there are enough triplets to code for each of the different possible amino acids. Okay, so we've covered DNA, we've covered its function. DNA is divided up into genes, and before each gene, so we'll say this is a gene here. Before each gene, you have a gene regulatory sequence, and this is where the gene regulatory proteins sit. And these gene regulatory proteins then decide when this gene will get read and when it will get copied, and therefore when it will be used to create a new protein. So we're going to talk about another component of the cell, which is the organelles. And the organelles are kind of like the cell's organs. So we've talked about one of them already, the nucleus, which is the DNA storage house and often considered to be the brain of the cell. Another organelle that's important is the mitochondria. And mitochondria are normally drawn something like this. And this is because mitochondria have a double layer of membrane. So we have a membrane layer here, this is the cell membrane. We have a membrane layer here, this is the nucleus's membrane. And we have 2 membrane layers here. These are the inner and outer membrane layer of the mitochondria. And the mitochondria are generally considered to be the factories of the cell. They're used to turn energy that the cell is able to get into a usable form that the cell can use to power different types of reactions. So that is the function of mitochondria. Another organelle that you will find in cells is the endoplasmic reticulum. And the endoplasmic reticulum extends from the cell nucleus and often looks something like this. And the purpose of the endoplasmic reticulum is to produce proteins that are going to be leaving the cell, or that are going to be existing in organelles inside the cell. So we have proteins that are produced here in this area, outside the organelles, but inside the cell, which is known as the cytosol. And these proteins fulfill functions here in the cytosol. We will then also have proteins that are produced inside this endoplasmic reticulum, and those are proteins that are going to be leaving the cell, or that are going to be existing in an organelle inside the cell. So proteins are produced in the endoplasmic reticulum, and then they're passed on into a structure called the Golgi apparatus. And the Golgi apparatus is generally drawn something like this. And so proteins that are going to be leaving the cell will be produced in the endoplasmic reticulum. They'll move through into the endoplasmic reticulum, into the Golgi apparatus, and then on into these structures called vacuoles, which are just small spheres with a phospholipid membrane that can then fuse with the cell membrane. We'll zoom in here to see how that can look. The vacuole will fuse with the cell membrane. And when it does that, its contents will be released outside the cell. So there can also, objects can also be taken in through vacuoles in the opposite way. So you have a vacuole forming in the cell membrane. Objects end up inside this vacuole, and then it becomes a separate vacuole inside the cell. Okay, so we have vesicles. Another organelle that's important to talk about is the lysosome. And sometimes when we've taken things into the cell, we want to break them down because we want to use the components to build objects inside the cell. So proteins will be broken down to amino acids, DNA will be broken into nucleotides, more complex sugars will be broken down into more simple sugars. And for this purpose, we have a structure called a lysosome. The lysosome is a bigger vacuole that is full of enzymes that will break down whatever ends up inside it. Okay, so now we've talked about the fundamentals of a cell. So a cell is, just to review, a cell is the lowest indivisible building block of life. And here, for example, we have an animal cell. It has a membrane composed of phospholipids. It has a lot of water, 50 to 70% of the cell's content is water molecules. It has proteins that fulfill lots of different functions. We have enzymes, we have structural components, we have membrane transporters, we have gene regulatory proteins. The cell also has nucleotides, which put together can form DNA, and DNA is the blueprint for how new proteins are going to be built. And the DNA is also the hereditary information that is passed on to the next generation when the cell divides. And in the cell, we will find different types of membrane-covered organelles. We have the nucleus, the endoplasmic reticulum, the Golgi apparatus, we have vacuoles, we have mitochondria, and we have lysosomes. So that is the basics of what a cell is made up of. Thank you for listening.